In the last video, we discussed the structural differences between DNA and RNA and the functional implications of those differences. The topic of this video is the role of base pairing hydrogen bond interactions in the central dogma of biology. And so the central dogma of biology is the set of three fundamental ideas in biology. And the first two of those are that DNA contains genes. So basically, DNA is broken up into shorter segments, and those shorter segments encode for an RNA sequence. And that RNA sequence encodes for an amino acid sequence. Okay? So what this is saying is that the sequence of the DNA molecule determines the sequence of the RNA molecule, specifically the messenger RNA or mRNA molecule, which in turn determines the amino acid sequence of the protein that's being made. And we'll talk in great detail later in the semester about how that specific amino acid sequence encoded by the DNA and the RNA is essential to determining how the protein will end up folding and what structure that protein will end up adopting, which is then essential to determining what function that protein will have. So how does this actually work? So the process where DNA is converted to RNA is called transcription. You can think of this as DNA and RNA, they're both made up of nucleotides, so they speak the same language. So it's kind of like the cell is just writing down a note when they're transcribing DNA and the RNA. And they can take that note to another location to actually build the protein that is being encoded by the DNA. To do that, that mRNA molecule, that note that we wrote down, is translated from the language of nucleic acids and nucleotides into the language of proteins and amino acids. So those are the first two concepts included in the central dogma of biology. The last one is that DNA actually directs its own replication. And this occurs during cell division to ensure that both daughter cells end up with the proper amount of DNA. And the key idea here is that all three of these processes are driven by the same thing. They're all directed by our base pairing rules, which determine which base pairs are complementary to each other. So let's look at this in a little bit more detail. So we know that the main function of DNA is to act as a long-term storage molecule for the genetic information and to be very stable. But going along with its function as the storage molecule, it also has to be able to replicate so that during cell division, both daughter cells end up with the right amount of DNA. And it turns out that this process is entirely self-directed. So how does this work? The way that this works is that because DNA is double-stranded, one of these strands can act as a template for the production of the new strands. And so a couple other key words are that the template strand is also called the parental strand, and the new strand is also called the daughter strand. Okay, and the reason that this is possible is because of our base pairing rules, which say that in DNA, no matter what, adenine is base pairing with thymine, and guanine is base pairing with cytosine. So what this actually means is that the nucleotide that needs to be added to the new DNA strand, the new daughter strand, is already predetermined by whatever nucleotide is present in the template strand. Okay, so here we have a cytosine, so it's predetermined that the daughter strand has to have a guanine at that position. All right, So the base pairing rules basically dictate what nucleotide has to be inserted where, which allows DNA to act as a template for its own replication. The same idea is true of transcription. So as the genetic storage molecule, DNA also has to be accessible to have those instructions converted into a usable form. That's transcription. This works in the exact same way. This is the same thing, base pairing rules. Because DNA and RNA are both nucleic acids, the ribonucleotides can still base pair with the deoxynucleotides in DNA. And so we're undergoing the same process where the DNA template, whatever nucleotide is present there, so in this case adenine, determines what nucleotide needs to be added to the messenger RNA. So in this case, we would need uracil. And that is the only difference between DNA replication and transcription, really, is that instead of base pairing with thymine, adenine base pairs with uracil, guanine still base pairs with cytosine. And the base pairing rules are still dictating what nucleotide needs to be inserted where. So that's replication and transcription. Translation is actually very, very similar, even though we're going from nucleic acid, the protein, and it's happening in the ribosome. So the way that translation works is that the messenger RNA can be broken up into 3 nucleotide, or you might see nucleotide noted as NT. That just means nucleotide. Words. So 3 nucleotide words that are typically called codons. And those are on the messenger RNA. So we have these sets of 3 nucleotides. And each one of these codons has a complementary anticodon on a tRNA molecule. And the tRNA molecule has this anticodon, and it also has an amino acid attached to it. So in this way, we're still having base pairing driving protein synthesis. So what has to happen is that the correct anticodon that has the right base pairing has to match up with the codon on the mRNA. When that correct anticodon matches up with the codon, it brings the correct amino acid to the ribosome. That correct amino acid can then be added to the growing peptide chain, and we have protein synthesis occurring. So this is again all driven by the correct base pairing interactions. But what is actually happening? How does the cell know that the correct base pairing interaction is happening? So it's actually the same in all 3 of these cases. So what it comes down to is that each of these processes has a protein or an RNA molecule that is able to interact with that minor groove on the DNA, mRNA, tRNA to determine correct base pairing geometry. So what's happening is that, let's say we're talking about DNA replication. So we're making this new strand of DNA, and when the correct base pair is formed, it results in a very specific geometry of that base pair. the bases have a specific geometry. And this geometry is only possible if you have the correct base pairing interactions. So what needs to happen is that these hydrogen bond acceptors I'm circling here have to end up in those exact positions. There's very specific positions that they have to be in order to have the proper interactions with the proteins involved in these processes. And this positioning is only possible with the correct base pairing. Without the correct base pairing pattern, we can't have the proper interactions forming with the proteins involved in these processes. And so that building block, that nucleotide or amino acid, will not end up being added because it is incorrect. And this is all coming down to the fact that only these correct base pairing interactions can produce this optimal geometry where our hydrogen bond acceptors are placed where they need to be. And so that is how base pairing is actually playing an essential part of allowing DNA replication, transcription, and translation to occur properly to make the right proteins. For the next video, I'm going to have you watch videos about two different types of DNA sequencing methods. So the original method, which is called Sanger sequencing, as well as the modern version of that, and then a completely different method called next-gen sequencing or Illumina sequencing.